Implementation of tardigrade protein synthesis in 2024, a comprehensive report. Overview. In 2020, the bioengineering sector stands on the brink of a revolutionary advancement with the implementation of tardigrade protein synthesis driven by a strategic investment of $3.5 million aimed at enhancing bioengineering facilities. Tardigrades, colloquially known as water bears, are renowned for their extraordinary resilience and adaptability, possessing unique proteins that enable them to thrive in extreme environments. This initiative is expected to unlock innovative applications across various fields including medicine, agriculture, and environmental science. Key steps for implementation. 1. Research and development. The first critical step in this implementation involves extensive research dedicated to understanding the specific proteins synthesized by tardigrades. These proteins are key to their survival in extreme conditions such as high radiation, extreme temperatures, and desiccation. Researchers aim to elucidate the biochemical pathways and molecular mechanisms that confer this resilience. Laying the groundwork for practical applications in various sectors. Collaborations with academic institutions and research organizations are being established to expedite this foundational research. 2. Genetic engineering. Following the detailed research phase, the next step involves employing advanced genetic engineering techniques to replicate or modify tardigrade proteins for utilization in other organisms. Techniques such as CRISPR-Cas9 gene editing will be pivotal in integrating tardigrade genes into crops, thereby enhancing their resistance to environmental stresses like drought, pests, and diseases. This genetic modification could lead to the development of hardier agricultural varieties that can thrive in adverse conditions, significantly improving food security. 3. Pilot testing. The third phase will focus on conducting rigorous pilot tests in controlled environments to evaluate the efficacy of the engineered proteins. These tests will assess the performance, viability, and ecological impacts of modified organisms in real-world scenarios. Data collected during this phase will be crucial for refining the genetic engineering processes and ensuring that the applications are safe and effective before scaling up production. 4. Scaling up production. Upon successful completion of pilot tests, the final step involves scaling up production processes to ensure that bioengineering facilities can produce the modified organisms or proteins at a commercial level. This phase will require investment in infrastructure, technology, and trained personnel to meet the anticipated demand for these innovative bioengineered solutions. The scaling up process aims to create a sustainable model for the production and distribution of engineered crops and proteins. Investment and economic implications. The $3.5 million investment is not solely focused on research but also aims to upgrade existing bioengineering facilities to accommodate new technologies and processes. This financial commitment reflects a growing recognition of the potential economic benefits of bioengineering innovations, particularly in enhancing agricultural productivity and developing sustainable practices. The investment is expected to create jobs, stimulate economic growth, and position the investing entities as leaders in the burgeoning field of bioengineering. Common themes across sources. Unique properties of tardigrade proteins. The emphasis on the unique properties of tardigrade proteins highlights their potential to revolutionize various industries, especially agriculture. Applications in agriculture. There is a consensus on the potential applications of this research in enhancing crop resilience and food security. Importance of funding and resources. All sources underline the critical role of funding and resources in driving these initiatives forward, suggesting that financial backing is essential for success. Differences between sources. Focus areas. While some sources emphasize agricultural applications, others highlight the potential uses in pharmaceuticals and environmental remediation, indicating a broader interest in the implications of tardigrade protein synthesis. Methodologies and timelines. The timeline and specific methodologies for genetic engineering vary slightly among sources, indicating differences in research approaches or institutional focuses. Timeline of events. Early 2024, initial funding was announced leading to the formation of specialized research teams dedicated to studying tardigrade proteins. Mid-2024, completion of foundational research into the biochemical properties and synthesis pathways of tardigrade proteins. Late 2024, initiation of genetic engineering trials, with preliminary results expected to be published by the end of the year. 2025, pending successful pilot test outcomes, scaling up production will commence, paving the way for commercial applications in agriculture and beyond. Conclusion. The integration of tardigrade protein synthesis into bioengineering represents a promising frontier for innovative applications supported by substantial investments and a structured implementation process. The collective efforts in research, genetic engineering, and pilot testing are poised to yield significant advancements in resilience and sustainability across multiple sectors. As this initiative unfolds, the potential benefits for agriculture, medicine, and environmental science will contribute to a more sustainable and resilient future, addressing some of the most pressing challenges faced by humanity today. The content presents a forward-looking initiative in bioengineering concerning tardigrade protein synthesis. Here are some counterintuitive pieces of information within the provided material. 1. Tardigrades' common perception versus their potential. Tardigrades, often dismissed as mere microscopic creatures, are commonly known for their ability to withstand extreme conditions such as high radiation, extreme temperatures, and even the vacuum of space. However, the counterintuitive aspect is that these resilient traits can be harnessed not just for theoretical applications but for practical advancements in agriculture and medicine. The idea that such a small organism can have a profound impact on human technology and sustainability challenges the often held belief that larger organisms or more complex systems are inherently more valuable for bioengineering. 2. Investment in bioengineering facilities. The announcement of a $3.5 million investment may seem modest compared to the scale of biological research in other fields such as pharmaceuticals or advanced material sciences. This could imply that bioengineering, particularly focused on natural organisms like tardigrades, is still considered a niche area. Despite its potential, the counterintuitive takeaway is that significant breakthroughs can arise from relatively small investments in emerging technologies, suggesting that the future of bioengineering may not require vast amounts of funding to yield transformative results. 3. Genetic engineering's role. The use of CRISPR technology for integrating tardigrade genes into crops may seem straightforward, yet the complexity of genetic engineering in agriculture often leads to skepticism about its feasibility and safety. People might assume that genetic modifications lead to unpredictable results. The counterintuitive point here is that the very precision of CRISPR technology can lead to more predictable and controlled outcomes, potentially making crops more resilient rather than causing adverse effects, which is a common fear associated with genetically modified organisms, GMOs. For pilot testing phase, the third step in the implementation plan emphasizes pilot testing in controlled environments. It's somewhat counterintuitive that rather than immediately rolling out the technology for widespread use, the focus is on thorough testing first. In many industries, rapid deployment is favored over cautious testing due to market pressures. However, this initiative suggests that careful assessment and data collection can lead to more robust and effective applications, ultimately benefiting the bioengineering field by establishing trust and reliability. 5. Economic recognition of bioengineering innovations. The economic implications of the $3.5 million investment suggest a growing recognition of bioengineering's potential. Counterintuitively, while many industries have historically relied on traditional methods, the push towards bioengineering advancements indicates a shift in thinking. This could lead to more sustainable and efficient practices. Though the established agricultural and pharmaceutical industries may resist such changes due to the perceived risks involved with new technologies, 6. Diverse applications beyond agriculture. While the content primarily discusses agricultural applications, it notes potential uses in pharmaceuticals and environmental remediation as well. The counterintuitive notion is that a focus on a single organism, tardigrades, could lead to a wide array of applications across distinct fields. This challenges the common belief that specialized research yields limited outcomes, highlighting instead that interdisciplinary approaches can open new avenues for innovation. These insights reflect a broader trend of re-evaluating the potential of seemingly simple biological mechanisms and their implications for future technological advancements in various sectors. The report on the implementation of tardigrade protein synthesis in 2024 highlights several critical aspects that may not receive the attention they deserve. Yet they are significant for a comprehensive understanding of the initiative's potential impact. Overlooked aspects of the report. 1. Mechanisms of tardigrade resilience. Many readers may focus on the application of tardigrade proteins without fully grasping the underlying biological mechanisms that confer resilience to these organisms. Tardigrades can survive extreme temperatures, radiation, dehydration, and even the vacuum of space due to unique proteins such as tritolose and protective proteins that stabilize their cells. Understanding these mechanisms is crucial because it not only informs the genetic engineering process but also influences the scope of applications in agriculture and medicine. For instance, if researchers can isolate specific proteins responsible for desiccation tolerance, these could be integrated into crop species to enhance drought resistance. Ignoring the foundational biology could lead to incomplete applications or unforeseen consequences in bioengineering. 2. Ethical and environmental considerations. The report may not emphasize the ethical implications of genetic engineering and the environmental impacts of introducing modified organisms into ecosystems. The potential for gene flow into wild populations, unanticipated ecological interactions, and the long-term consequences of deploying genetically modified crops are significant concerns. Addressing these ethical and environmental considerations is essential for gaining public acceptance and ensuring that bioengineering advancements do not result in ecological harm. This aspect is often overlooked in discussions focused solely on technological benefits, yet it could impede the implementation of these innovations if not adequately addressed. 3. Economic investment justification. While the report mentions a $3.5 million investment, it does not delve into the rationale behind this funding or how it will be allocated. Understanding whether this investment is primarily for research facility upgrades or workforce training is crucial. Furthermore, the report lacks a discussion on the anticipated return on investment (ROI) or economic benefits projected from successful applications of tardigrade protein synthesis. This information is vital for stakeholders, investors, and policymakers to assess the viability of the initiative and its potential for driving economic growth in the bioengineering sector. 4. Interdisciplinary collaboration. The implementation of tardigrade protein synthesis will require collaboration across various scientific disciplines, including molecular biology, genetics, agronomy, and environmental science. However, the report does not highlight how interdisciplinary approaches will be facilitated or encouraged within the research teams. Effective collaboration could enhance innovation and lead to more robust solutions, but without a structured framework for interdisciplinary work, the initiative may face challenges in integrating diverse expertise and perspectives. 5. Public perception and engagement. There is little mention of how public perception and stakeholder engagement will be managed throughout the implementation process. Public acceptance of genetically modified organisms (GMOs) can significantly influence the success of bioengineering projects. Engaging with communities, addressing concerns, and providing transparent information about the risks and benefits of using tardigrade proteins is essential for building trust. Failure to consider public sentiment could lead to resistance, potential regulatory hurdles, and delayed timelines for bringing products to market. Significance of these overlooked aspects. The significance of addressing these overlooked elements cannot be overstated. Scientific foundation: a deep understanding of tardigrade biology is paramount for effective application. The more researchers know about the proteins involved in resilience, the better equipped they will be to engineer crops and other organisms that can withstand environmental stressors. Ethics and sustainability: as society grapples with climate change and food security, ethical considerations in bioengineering cannot be sidelined. Ensuring that advancements do not harm ecological systems aligns with broader sustainability goals and fosters public support. Investment clarity: clear justification for financial investments can attract further funding and support from governmental and private sectors. Demonstrating a clear ROI can also encourage similar initiatives in the future. Collaboration as a catalyst for innovation: interdisciplinary collaboration can lead to groundbreaking discoveries that might not emerge from isolated research efforts. Encouraging cooperation among scientists from different fields can yield innovative solutions. Public trust and acceptance: engaging with the public and addressing their concerns will be crucial for the long-term success of genetic engineering projects. Building trust can facilitate smoother introductions of new technologies into the market. In conclusion, while the report on tardigrade protein synthesis provides a clear roadmap for implementation, the significance of the overlooked aspects discussed above is crucial for ensuring the success and acceptance of this initiative. A balanced approach that integrates scientific understanding, ethical considerations, economic justification, interdisciplinary collaboration, and public engagement will be essential for harnessing the full potential of tardigrade proteins in bioengineering. 1. Research and development of tardigrade proteins. The first and foremost step in the implementation of tardigrade protein synthesis is extensive research into the unique proteins synthesized by tardigrades that contribute to their remarkable resilience in extreme conditions. This foundational research is critical as it informs subsequent steps in the development process. Understanding the biochemical pathways, structural properties, and functional roles of these proteins can unlock potential applications across various sectors, including agriculture, medicine, and environmental science. 2. Genetic engineering techniques. The second key point involves the application of advanced genetic engineering methods, particularly CRISPR technology, to replicate or modify tardigrade proteins for use in other organisms. This step is crucial for enhancing the resilience of crops and developing new biotechnological applications. By integrating tardigrade genes into plants, researchers aim to create strains that are more resistant to environmental stresses like drought, pests, and diseases. The implications of this step are far-reaching, as it could lead to significant improvements in agricultural productivity and sustainability. 3. Pilot testing and evaluation. The third step emphasizes the importance of pilot testing engineered organisms or proteins in controlled environments. This phase is designed to evaluate the efficacy and viability of the engineered solutions before moving towards large-scale production. Gathering data on performance and real-world applicability is essential to ensure that the innovations can meet the desired outcomes. This step serves as a critical checkpoint to refine the approaches and ensure that the applications can withstand practical challenges. 4. Economic implications and investment in facilities. The $3.5 million investment in bioengineering facilities represents a significant commitment to advancing this field. This funding is not only intended for research but also for upgrading existing facilities to accommodate new technologies and processes. The economic implications of this initiative are profound, as successful implementation of tardigrade protein synthesis could lead to enhanced crop yields, sustainable agricultural practices, and new biotechnological products. This investment reflects a broader recognition of the potential benefits of bioengineering innovations, potentially driving economic growth and addressing global challenges in food security and sustainability. 1. The Human Genome Project 1990 to 2003. This groundbreaking international scientific research initiative aimed to map all the genes of the human species. With an investment of approximately $3 billion, it involved collaboration among multiple countries and institutions. The project not only provided a complete reference for human DNA but also paved the way for advances in genetic research and biotechnology similar to the Tardigrade protein synthesis initiative that seeks to harness genetic engineering for practical applications. 2. CRISPR-Cas9 gene editing 2012-present. The development and implementation of CRISPR technology has revolutionized genetic engineering. With its ability to edit genes with precision and efficiency, CRISPR has resulted in various applications, including agriculture, medicine, and research. The significant investment in research and development to explore CRISPR's potential mirrors the investment in tardigrade protein synthesis, indicating a broader trend in biotechnology to utilize genetic modifications for enhancing resilience and adaptability in various organisms. 3. Transgenic crops development 1994-present. The introduction of genetically modified organisms (GMOs), particularly transgenic crops engineered for pest resistance and herbicide tolerance, represents a significant advancement in agricultural biotechnology. This movement began in the early 1990s and has since grown into a multi-billion-dollar industry. The focus on enhancing crop resilience through genetic modification is parallel to the goals of the Tardigrade protein synthesis initiative, which aims to use genetic engineering to improve agricultural practices and sustainability. These historical cases illustrate the transformative potential of biotechnology and genetic engineering in various sectors. Echoing the ambitions of the current Tardigrade protein synthesis project. Future outlook: Tardigrade protein synthesis and bioengineering advancements. The year 2024 marks a pivotal moment for the bioengineering sector with the anticipated implementation of tardigrade protein synthesis backed by a strategic investment of $3.5 million aimed at enhancing bioengineering facilities. This initiative harnesses the unique properties of tardigrades, known for their extraordinary resilience to extreme environmental conditions, to foster innovative applications across various fields including agriculture, medicine, and environmental science. Key steps for implementation. 1. Research and development. The foundational step involves comprehensive research focused on identifying the specific proteins synthesized by tardigrades that confer their remarkable survival abilities. Detailed investigations into the molecular mechanisms and genetic pathways responsible for this resilience are crucial for translating these findings into practical applications that can benefit humanity. 2. Genetic engineering. Following the research phase, the next step will involve advanced genetic engineering techniques to replicate or modify these proteins for application in other organisms. Techniques such as CRISPR will play a significant role in integrating tardigrade genes into crops, enhancing their resistance to environmental stresses like drought and pests. This genetic manipulation could revolutionize agricultural practices by developing crops that can thrive in adverse conditions. 3. Pilot testing. The third phase will focus on pilot testing in controlled environments to evaluate the effectiveness and viability of the engineered proteins. These trials will provide critical data on performance metrics, thereby assessing how these advancements can be applied in real-world agricultural settings and beyond. 4. Scaling up production. Upon successful pilot tests, the project will transition into scaling up production processes. This step is essential to ensure that bioengineering facilities can produce modified organisms or proteins at a commercial level, paving the way for widespread application and utilization in various sectors. Investment and economic implications. The $3.5 million investment is strategically aimed at not only funding research but also upgrading existing bioengineering facilities to incorporate new technologies and processes. This financial commitment reflects a growing recognition of the economic potential inherent in bioengineering innovations, particularly in enhancing agricultural productivity and promoting sustainable practices. Predicted applications and trends. Agriculture. The most immediate applications of tardigrade protein synthesis are likely to be in the agricultural sector. Enhanced crop resilience can lead to improved yields, reduced dependency on chemical pesticides, and a more sustainable approach to food production. As climate change continues to challenge traditional farming methods, these innovations could provide critical solutions to ensure food security. Medicine. Beyond agriculture, the applications of tardigrade proteins may extend into medicine. Research may uncover therapeutic uses for these proteins in drug development, particularly in areas such as regenerative medicine, where the ability to withstand extreme conditions may translate into enhanced healing properties for human tissues. Environmental science. The potential for tardigrade proteins to contribute to environmental remediation efforts is another exciting avenue. These proteins could be engineered to aid in bioremediation processes, helping to clean up contaminated environments or improving the resilience of ecosystems facing climate change impacts. Economic growth. The advancements in bioengineering facilitated by this initiative are expected to stimulate economic growth. By fostering innovation, job creation in the biotech sector, and the development of new markets for engineered crops and therapeutic applications, the investment will likely yield substantial returns in the coming years. Common themes and divergent perspectives. Across various sources discussing this initiative, there is a consensus on the transformative potential of tardigrade proteins in revolutionizing industries, particularly agriculture. However, perspectives diverge on the timeline and specific methodologies for genetic engineering, reflecting varying research focuses. Some emphasize agricultural applications, while others highlight potential therapeutic uses and environmental benefits, indicating a broad spectrum of possible impacts. Timeline of events. Early 2024, announcement of initial funding leads to the formation of dedicated research teams. Mid-2024, completion of foundational research into the properties and mechanisms of tardigrade proteins. Late 2024, initiation of genetic engineering trials with preliminary results expected by year-end. 2025, if pilot tests are successful, scaling up production will commence, setting the stage for commercial applications. Conclusion. The integration of tardigrade protein synthesis into bioengineering represents a promising frontier for innovative applications across multiple sectors. The structured implementation process involving research, genetic engineering, pilot testing, and scaling up production is poised to yield significant advancements in resilience and sustainability, as the investment catalyzes research and development. The future of bioengineering holds the potential to address critical challenges in agriculture, medicine, and environmental sustainability, ultimately contributing to a more resilient and sustainable world.